Welcome back. Hopefully you guys are having a very good summer break. Mine is not going too bad. I will be trying, as you will hopefully see by the uploads on the YouTube channel, to try to upload a lot of these lectures as fast as I can. The audio might be a little bit different. I am doing this from the road. I'm not doing this like I normally do from my office, but without further ado, let's go ahead and get started. So last time we talked about acids and bases, pHs, how to do different calculations with that. This time we're going to start talking about something that's very important in biochemistry, and that's amino acids. Right? So amino acids are very important. They're so named because they have an amino group, as we'll see. And they have a carboxylic acid group. What we'll see is these two groups are very important for their chemistry and the way that they can bind to make a peptide bond. There are 20 naturally occurring amino acids. We will be using the L series. In their protein is about 4 dietary calories per gram. So of the 20 amino acids, 10 are known as essential. And what that means is that we need to eat them in our diet, that our body does not produce them. I'm not going to go through and draw all 20 amino acids. I just don't have the time. You can easily look it up online or in an MCAT study book or your textbook to see. The easiest way to study these amino acids and to memorize them is to just memorize the R group. This backbone does not change. The only slight variation is an amino acid called proline, where proline will fold back on itself, but it still has an R group and the backbone. And so all you have to do is memorize the backbone and then memorize the R group, and then that way you will be able to memorize all 20 amino acids relatively easily. So here is a mnemonic to help us know which of the 20 from the 20, which 10 are essential. And so we have phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, and then we have histidine, arginine, lysine, and leucine. Now, I will tell you this. We are not using the one-letter abbreviation codes for these amino acids, right? Arginine is R. Lysine is K. Phenylalanine is F. But to make sure that this mnemonic will hold, we're going to go ahead and just use the first letters. So we have phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, lysine, and leucine. Private Tim Hall. Those are the 10 essential amino acids. I will tell you arginine is only essential for nitrogen balance, for the way that nitrogen is passed through when we talk about nitrogen metabolism. So arginine is important in there. And arginine actually stops being essential, quote unquote, after as you start to grow. I believe it's about 10 years. Don't quote me on that. But those are the 10 essential ones. We need those from diets. You'll see that a lot of the aromatic amino acids we need from our diet. Plants make them, so eat your plants. Another thing we can notice about this amino acid is depending on the R group, this carbon is a chiral center. We're not going to go into much of a detail about chiral centers, and so what all we're going to talk about is the way that the amino acid is drawn here, whether you have the carboxylic here or you can put it up here where this hydrogen is. As long as this nitrogen group is on the left side, that denotes the L form. and L amino acids are the only ones that we're going to talk about. L does not talk about how light is rotated, if at all, like you might normally talk about S and R with your chiral centers. We're not going to worry about that. We use the L. We don't really know why the L form amino acid is naturally used. Some bacteria actually use the D form just to kind of throw us off. The other thing to notice with this amino acid, the way I drew it was at pH of 7. So this proton on this carboxylic acid has been deprotonated, and we have added a proton to the amino end. And so you can notice we have a positively charged amino group, a negatively charged carboxylic acid group, and this is known as a zwitterion. Right? A zwitterion is something that a molecule that has both a positive and negative charge in the same molecule. So, what can we do with these amino acids? So these amino acids, forgot to erase that. These amino acids can be linked to make proteins. And so let's look at an amino acid. And so I'm going to draw an amino acid, aspartate. Right, so there's aspartate. So D is the one-letter code for aspartate. We add heat, and then we're going to add another one. We're going to add alanine. Right, so alanine is a pretty simple amino acid. And by simple, I mean R groups. So this is alanine. And so what we can do is with a little bit of heat, we can combine these two together. And what I'm going to do is just focus on the peptide bond. And what we're going to do is we'll talk about ways that these things come together and what they actually can do. So, here's our peptide. We have aspartyl alanine, right? Aspartyl alanine. This would be known as a dipeptide. It's a dipeptide caused by a peptide bond between two amino acids. The other thing that we notice is I'm actually going to draw this amino group slightly differently. I'm going to draw it like this. Because one of the things that's very important is the formation of a peptide bond is a dehydration reaction. So we're going to lose H2O, right? So then the easiest way to break a peptide bond is to add a little bit of heat and add water back. And that'll help us break that peptide bond. So here we have this dipeptide. We have a directionality formed with these proteins. And this is known as the N-terminus, which is the free amino group. This is known as the C-terminus, right? The free carboxylic acid group. And so what you can see is that it goes N-term to C-term. So every time we add a subsequent amino acid, it will be added to the C-terminus. And so just like DNA goes from 5' to 3', proteins or peptides go from N-terminus to C-terminus. Here is the peptide bond. That right there is the peptide bond. And so if we were to be ever asked to draw a larger peptide bond than this, there's a little tip. There's a little hint. There's a little thing that we can use to make sure that we drew it correctly. And that it goes N, C, C double bond O. N, C, C double bond O. Whether it's in the peptide bond or it's free, it always goes N, C, C double bond O. That first C is always known as the alpha carbon, right? As I drew up here, this alpha carbon has the hydrogen attached to it as well as the R group. And so it goes N, C, C double bond O. N, C, C double bond O. If you can just get the backbone right, then all you would have to do is draw the R group, the correct R groups for that peptide chain. Now, I drew this aspartate, and then alanine. But since we're doing this just in a beaker, I'm going to go ahead and get a new slide. Couldn't we also make this? So N, C, C double bond O, N, C, C double bond O. Right? So there's our pattern. I could also do this. So if you remember before, and I'll go back, oh, not there. And I'll go back to show you guys again. So before, we made aspartyl alanine, right? So aspartate is there, alanine is there, but couldn't we also make alanine or alanyl aspartate? And the correct answer is yes. When you just have two peptides, or when you have a variety of different amino acids just in solution with a little bit of heat, you can make a variety of different combinations. Now, not only can we make combinations from the N-terminus, right? N-terminus to the C-terminus, but we can also use R groups depending on where we are, right? And what R group is present. So let's look at the R group of aspartate. Man, that awfully looks like a C-terminus. So in the case of this, when we're just putting amino acids in solution, some R groups can form a peptide bond. And so what we actually get formed is this. So this is going to be something, so these are the three things that would be formed in a beaker if I put aspartate and alanine in there. And so it's just something to keep in mind. Whoops. So we're making a peptide bond. So there's the N. And I'm going to draw this slightly up because of the space. So let's go ahead and circle. I'm going to circle the alpha carbons in red. There's an alpha carbon. There's an alpha carbon. Right, so if this is the alpha carbon, if this is the alpha carbon, that is 2, then this is going to be the beta carbon. If that's the beta carbon, then this is the gamma carbon. Right, and so this would be gamma aspartyl alanine, right? Depending on how it binds, right? And so we can name it. Normally, we don't have to put the alpha, aspartyl alanine or alpha alanyl aspartate. And so this way, we can just be specific. So just some nomenclature things. Oligopeptides. So oligopeptides. So let me draw the O. Are about 10 amino acids in length. If you have less than 10, you're known as a polypeptide. Or sorry, if you're greater than 10, you're a polypeptide. If you're greater than 20, you're usually known as a protein. And so this will come back later on as we discuss protein formation and function, and a variety of things like that. So let's talk. And this will be the last thing we talk about, not this. Let's talk about titrations of amino acids, right? So we talked a little bit about this when we talked about amino acids and histidine and why do we care. And so let's draw glycine. So let's look at, we'll start at the simplest, and then maybe we'll go on to a polypeptide. And so the way we do this is we draw everything as in its fully protonated form. So here's glycine fully protonated. Right? We have a pKa. We'll call this pK1 of, let's do a circle in pink, of this proton right there. Right? So when we go above that pKa, we remove that proton. So above pKa's, we remove protons. Below pKa's, we add protons. There are a variety of different pKa tables that you guys can look at about the different amino acids, because it will change depending on the N-terminus, the C-terminus, as well as the R groups. And so then what we can have is a pK2. for this amino group. And above it, we're going to take it off. Whoops, I don't know why I did that. My apologies. We can take it off. And so what we have is we have a +1 total charge, we have a 0 total charge, and a -1 total charge. There's a point known as the isoelectric point. We can calculate isoelectric points for amino acids as well as for peptides. The isoelectric point is the point where there's no net charge. Right there. And so when there's no net charge, what you can do to find the pi is take pK1 plus pK2 divided by 2. Now these pKs will surround the 0 charged state. This might be different depending on which amino acid you will look. My recommendation would just to try it on aspartate. We looked at aspartate before. Try it on aspartate, or try it on glutamates, or try it on these other lysines, just so that you can get the hang of, okay, these are the 2 pKa's that I need to use to find the pi. Now, I'm showing you this in the sense of just a single, wow, I forgot to erase these before, from the single amino acid. Really, let's look at this in the context of a peptide. Now, I'm going to show you an even number peptide, or oligotide. Or oligoprotein, excuse me. And so you can extend this out. You can make this even odd. Odd numbers are a little bit more difficult because you have to start playing with charges and looking at pKa's. Just remember, above a pKa, they lose a proton. Below a pKa, they gain a proton. So let's go ahead and draw. Whoops. Let's go and draw this. So we have alanine, glutamate, glycine, and lysine. And I want you to calculate the pi, the isoelectric point, of this tetrapeptide. So, NC, C double bond O, NC, C double bond O, N, C, C double bond O, and then one more, N, C, C double bond O. All right. So once we get that, now we can do the R groups. Glutamate is like aspartate, only an extra carbon. Normally when we draw these peptides, we're going to draw them out as pH of 7. So I'm going to draw the charges as if they were pH of 7. Ooh, might not have enough room for this. Yep, I got it. Okay, so now it's time. So the first step in calculating the pi. So what's the pi of this tetrapeptide? Okay, so we need all the pKa's. So we go to our pKa table, and really the pi is done neutral. Doesn't matter the pH. I'm drawing this as a pH of 7 because there's usually a follow-up question after this pi question, and so and we'll explain it there. So let's look at this. So nitrogen or the hydrogen of an amino group, it's usually around pH of 9 to 10. When we go to the pKa table, it'll be 9.69. I'm calling this pK2 because normally tables will call that pK2. The hydrogen from this carboxylic group is usually around a 2 to a 3. Call it pK1. It's 2.18. Okay, this right here, since it's in our group, is a little bit, we'll do pKr1. It's a little bit bigger at 3.22. And then this proton, we'll call it pKr2, is 10.53. Okay, so first thing we do is we look at everything that has a pKa and we write it down. The second thing we do is we then order these from lowest to highest, and lowest pH being the smallest number. So we have 1, 2, 3, 4. In this example, we only have 4 pKa's. Awesome. What we now do is we can take the middle 2, and that will be the pi. So we're going to take number 2, and we're going to take number 3. Okay? And the reason why we can do that is because if we go above number 2, this will be protonated and will be charged. we will have a +1 charge. If we go below or higher than this pK2, this will lose a proton and we'll have a -1 charge. So in between these two pKa's, we have a 0 charge. So let's do this. 9.69 plus 3.22 divided by 2, and we're going to get 6.46 as the PI, which makes sense. So at pH of 7, if we look at this, pH of 7 is really close to that, we should have a neutral charge. And we do -1, -1, +1, -1, +1. So +2 total, -2 total, we're a neutral peptide. So this is amino acids. What we're going to see next, in the next video, is we're going to see protein structure. We're going to see how we can start adding amino acids together to get proteins. And then the next section, the next video we'll talk about in this section will be how do we study those proteins? How do we study a protein that's in a cell?